Hello everyone, welcome back to my channel, and in this lecture we are going to particularly talk about the light reactions. Now you know that the photosynthesis process is the process in which the plant is producing sugar molecules and releasing oxygen in the presence of sunlight. So photosynthesis has been divided into two types of reactions. The first is the light reactions and the second is the dark reactions. Now, both of these light and dark reactions, they are happening on different sites of the chloroplasts. The light reactions are happening in the thylakoids when the light falls upon the plant. So thylakoids are basically the coin-like structures which are present inside the chloroplast. So let's suppose this is a chloroplast. This is a double membrane structure. Inside the chloroplast, there are these coin-like structures present, and each of this coin-like structure is individually called a thylakoid. And when these thylakoids stack onto each other, they are collectively known as the granum. And also, they are connected to each other, all the granum, through lamella. And the empty space, which is present in the chloroplast, that is filled with the fluid, which is known as the stroma. Now, these light reactions are happening in the thylakoids when the light falls on it. And in this process, water is split into oxygen and some electrons are being extracted, which go further down in the electron transport chain for the production of high energy molecules, particularly ATP and NADPH. Now, these end products from the light reaction, they enter into the stroma where the dark reactions, or you can call it as carbon reactions, take place for the production of sugar molecules. So this is how the photosynthetic process is taking place. And now we are going to look into detail the light reactions. Now, the light reactions are the reactions which are happening in the presence of light. And where are they happening? They are taking place in the thylakoid membrane. So here what you can see is the thylakoid membrane. So now this light reaction is basically a series of electron transport chain where some electrons are extracted from something and they are carried forward through a chain, and finally they are accepted by a final acceptor molecule for the production of high energy molecule. So that is what we are going to understand here. So the whole of the light reactions, in these reactions, there is the role of a very important system which is known as photosystems. What are they called? Photosystems. The name itself is suggesting that this is some kind of system which is having a relation with photons. And what are photons? Basically the component of light. So light has something to do with this photosystems. Now, photosystems, if I try to tell the literal meaning of this, these are the cluster of pigment molecules. So these are the cluster of pigments. So there are two photosystems present in the light reactions, or you can call it in the thylakoid membrane. There are two photosystems which are involved in the light reactions, and these are the photosystem 2 and the photosystem 1. Now, these two photosystems, they are cluster of pigments. What kind of pigments? So two kind of pigments. One is the primary pigment, and the other one are the accessory pigments. The primary pigment is the pigment which is having the main role in the electron transport chain, and that is the chlorophyll A molecules. On the other hand side, the accessory pigments are the pigments which are helping the primary pigment, that is chlorophyll A, in the absorption of the electrons or the light. And what are these accessory pigments called? These are the chlorophyll B molecules. These are the xanthophylls and also carotenoids. So these are the different accessory pigments which are helping the primary pigment in absorption of the light. Now, there is a reaction center present in the photosystem in the center, and this reaction center comprises of a special pair of chlorophyll A molecule. So there are two chlorophyll A molecules, special type of chlorophyll A molecules. They are forming a pair and they are present in the reaction center. For the electrons to reach this reaction center, the photons from the light, they are first absorbed by the accessory pigments, and then the light, the photons and the electrons, they are transferred to the reaction center, that is chlorophyll A molecule, where the electron transport chain starts. So I hope this thing is clear, that what is the role of primary pigment chlorophyll A and what is the role of accessory pigments. Okay, so now what happens is that the light falls onto these photosystems. Photosystem 2 is also known as P680. Why? Because the photosystem 2 is able to absorb 680 nanometer light at its best. So out of all the wavelengths, 680 nanometer light is the one that is absorbed maximally by PS2. On the other hand side, the PS1 is also known as P700, which means that it is able to absorb the light of 700 nanometers maximally. Now, what happens is that when light falls onto these two photosystems, the reaction center, which is having the chlorophyll A, they absorb the light eventually through the accessory pigments, and the electrons from that, they go into their higher energy state. So the electron you can see here, they go into their higher energy state, which means that they go into their excited state. Now, what happens when an electron goes into its excited state? There are two possibilities. Either the electron can make a bond with any other acceptor molecule, or this electron can be donated to the next acceptor molecule for it to come to the ground state. So this electron, the one that is excited in the photosystems, when it goes in the excitation state, they are accepted by another acceptor molecule which is sitting right beside the photosystem. And what is that? The first acceptor molecule here is the pheophytin. The pheophytin molecule is not depicted in the figure here, but you need to remember that. So pheophytin is the molecule or the acceptor molecule which is accepting the excited electron from the photosystem 2. Now the photosystem 2 is deprived of one electron. Just keep that in mind because we are going to come back to this theory. So the electron which is excited is accepted by pheophytin. Now this pheophytin is having extra electron. This electron will be now further donated to next acceptor molecule, which is known as the plastoquinone. Now plastoquinone, here you can see that this is the oxidized state of plastoquinone. It takes up the electrons and it goes into its reduced form, and hence now it is having more electrons. Now these electrons from the plastoquinone will be carried forward to the next acceptor molecule, and this acceptor molecule is called the cytochrome B6F. So cytochrome is the next acceptor molecule. After the cytochrome has gained the electron, now is the time to transfer it to the next molecule, next acceptor molecule. The next acceptor molecule in the series is known as the plastocyanin. Now, plastocyanin has received the electron. Where this electron is going, we will look to that after we have discussed what is happening at photosystem 1. So for now, just remember that photosystem 2, when light falls on it, there is an excitation of the electron. Electron goes into its excited state. This electron is then transferred to pheophytin, then it goes to plastoquinone, then it goes to cytochrome, and then it goes to plastocyanin. Now, what happens to the electron in the plastocyanin? We will come back to that. Now, let us discuss the story of photosystem 1. Now, photosystem 1 is again a cluster of pigments. It is also having the accessory pigments. It's also having the special pair of chlorophyll A molecules. So what is happening when the light falls? Again, electron is being excited. It goes into its excited state. This electron now will be donated to an acceptor molecule. And what is the acceptor molecule here? It is ferredoxin. So ferredoxin is the acceptor molecule here. And after the ferredoxin has gained the electron, it transfers that electron to the next acceptor molecule, which is ferredoxin reductase. Now, this is an enzyme. This is an enzyme which is going to do something. So this ferredoxin reductase is going to add the electrons to NADP and convert it into NADPH. So NADPH is now the reduced form. It has now gained the electrons. This is the high energy molecule, which is one of the end products of the light reactions. So this is here. So the photosystem 2 is the photosystem 1, the excited state electron is gone through a series of reactions, electron transport, and eventually it goes into NADPH. So at the end of the story, there is an electron deficient in PS2 and one electron deficient in PS1. So now we have to cover that up, right? We have to make sure that these electrons are gained back so that this process can keep on going. So now coming back to the story of plastocyanin, the electron that was delivered to plastocyanin through PS2, this is accepted by the photosystem 1 to regain the electron that it had transferred to NADPH. Now photosystem 1 is again back on its main state, right? And also there was an electron deficient in PS2 as well. From where are we going to get that electron and fill that gap? So that is being done by the splitting of water molecule. So the PS2, it extracts the electrons from the water molecule to fill up the gap of the electron it is now deficient in. And hence, by the splitting of the water molecule, oxygen is being produced, which is what we are breathing right now. So oxygen is a byproduct of the light reactions. And this process of splitting of the water molecule is known as photolysis. Now, when all this process is going on, what happens is that there is a proton gradient that is being produced. What I mean by this is that when the electrons, they are being transferred in the electron transport chain, some of the energy is being utilized for the transfer of the hydrogen ions from the stroma to the thylakoids. So there are an influx of hydrogen ions from the stroma to the thylakoid lumen. So this is happening when the electron transport chain was going on. So as the electron transport chain is going on, more and more hydrogen ions are gaining entry into the thylakoid, which is leading to an increase in the hydrogen ion concentration inside the thylakoid, which leads to an imbalance of the pH between the stroma and the lumen. Now, the concentration has changed. There is less concentration of hydrogen ions in the stroma, and there is more concentration of the hydrogen ions in the thylakoid. Now, we have to balance that out. That is how the homeostasis will be there. That is how equilibrium can be maintained. So in order to maintain the equilibrium, these hydrogen ions, they have to go back to the stroma so that an equilibrium can be achieved. How will that happen? There is no other way through the thylakoid membrane by which the hydrogen ions can move from the thylakoid lumen to the stroma, but there is one point from where this can happen, and that is because of one channel present inside a specific component, the enzyme ATP synthase, which is embedded in the thylakoid membrane. So what happens, all these protons, now the hydrogen ions which are accumulating inside the thylakoid, they go and gather near the ATP synthase molecule, and they start to move through the channel present in the ATP synthase so that the equilibrium can be maintained. Now, when these hydrogen ions, they are moving through the ATP synthase, some energy is being generated, some current is being generated, because of which the ADP molecules, they combine with the inorganic phosphate, which are present in excess in the stroma, and they combine it to form ATP molecules. Now, specifically, when 4 hydrogen ions are moving through the lumen, through the ATP synthase towards the stroma, one ATP molecule is being formed. So this is how the light reactions are taking place. So let us try to recapitulate everything and revise everything before we end this class. So the light reactions, they are taking place in the thylakoid the thylakoid membrane. Now, outside the thylakoid membrane is present the chloroplast stroma, and inside the thylakoid membrane is present the thylakoid lumen. There are 2 photosystems present inside the thylakoid membrane, which are participating in the light reactions. When light falls onto them, there is a series of electron transport chain happening where there is a final acceptor molecule present. When the light falls on PS2, the excited electron is accepted by pheophytin, then it is accepted by plastoquinone, then cytochrome, and finally to plastocyanin. Same story happens at photosystem 1, where the excited electron is accepted by the ferredoxin and then by ferredoxin reductase. And ferredoxin reductase is adding these electrons to NADP to reduce to its reduced form, that is NADPH, which is one of the end products. And the electron that is now deficient in the PS2 is accepted from the plastocyanin here. And the electron that is deficient in the PS2 is accepted from the splitting of water by the photolysis process. So this is how these things happen. And when the electron transport is happening, there is an influx of hydrogen ions from the stroma to the lumen. So subsequently, there is an increased concentration of the hydrogen ions inside the thylakoid, which is leading to a disruption of the equilibrium. And now to again gain back the equilibrium, these hydrogen ions, they have to go back to the stroma. How do they do that? By through a channel which is present inside the ATP synthase machinery, which is present in the thylakoid membrane itself. So now when 4 hydrogen ions from the thylakoid inside, they move through this channel, one ADP molecule and one PI molecule, that is the inorganic phosphate, they combine to each other to form ATP. So at the end of the light reactions, we are left with NADPH molecules and ATP molecules. Now these are present in the stroma and they will gain entry into the C3 cycle for the production of glucose molecules. So the splitting of water is known as photolysis. And this process where the hydrogen ions through the gradient, they are transferring to the stroma from the lumen for the production of ATP. This process is known as chemiosmosis. Also, because whole of this process looks like a Z, if you see here, this transport looks like a Z. That is why we also call it as Z scheme. So here in this figure, everything is very much clear and you guys can try to draw it in your notebooks in order to memorize it. So thank you so much.